So next up we have Marc Fuchs. He's from Cornell University. I met Marc down at the Sustainable Ag Expo in San Luis Obispo. I think it was 2017. And he gave 3 presentations at that Ag Expo. They were all fantastic, and he really simplified this whole complicated grapevine virus knowledge into really easy to understand concepts. And then he was so patient as I grilled him with questions during lunch, and he's been so supportive of our virus efforts in Lodi, really answering questions for us, and he came all the way from New York to be with us today. So please give a warm welcome to Professor Marc Fuchs.
Good morning, everyone. Thank you so much, Stephanie, for this wonderful introduction. My heartfelt gratitude is expressed to you and to every one of you sitting in the room today for taking time off your busy schedules and participating. And kudos to Stephanie and all of you for the wonderful program that you have launched to collectively try to mitigate the impact of these viruses. So, talking about my presentation, Stephanie tasks me today with the idea of communicating on what viruses are and to provide general information on their properties or some of their unique properties, because when you compare viruses to Pierce's disease or Xylella, the causal agent of Pierce's disease, other diseases that you might be more familiar with, like downy mildew, botrytis, rot, and so forth, viruses have very unique properties. So we're going to walk through these properties and explain you, based on these properties, how can one devise optimal management strategies. And these strategies, as you will see, are very, very simple. Actually, they are so simple that very often we neglect to consider them in a vineyard management program. So, this being said, let's go and start with viruses. What are viruses? Viruses are very small infectious agents. Their size is very, very, very tiny. They have different shapes. They have the genetic information that is quite unique, is very specific to a virus, meaning it can be either RNA or DNA based, and that's a different chemistry, but it can be very unique. And more importantly, not only are they tiny, they cause huge damage, but they're obligate parasites, meaning unlike bacteria and fungi, they can only live and survive on a living organism. The vine has to live in order for these infectious agents to thrive and multiply. So viruses are different shapes and sizes. This icosahedral shape here to the left is depicting grapevine fan leaf virus. The one to the right is depicting, which is a very filamentous shape, leaf roll. That's a particle of leaf roll virus. And in the middle here is a hypothetical representation of what red blotch should look like. And when I mention hypothetical, because nobody has seen red blotch particles in an electron microscope. So when you look at the size, I put scale bars here, they are very, very tiny, meaning that, for example, fan leaf has a diameter of about 10 nanometers, meaning that, to put everything in perspective, I'm about 6 foot tall, all right? If fan leaves were my size, my shoes would be in this room, and my head would be on the other side of the country on the East Coast, just to give you a perspective of how tiny these viruses are. And as I mentioned, they are very diverse in how they carry their genetic information. In other words, what is very key to them being able to survive after they infect a vine. Left again is fan leaf, and you see this icosahedral shape, so you can imagine roughly a basketball, and the ball inside contains the genetic information. For leaf roll, that is shown to your right-hand side, you have the genetic information that is twisted somehow along that spaghetti-like shape. And just in the middle, for the fun of it, I depicted another virus. This one does not go to grapes. It's a virus that infects bacteria, so a bacteriophage, and it's like coming from a Steven Spielberg movie, right? It looks like an alien, which in this head has the genetic information. So how do viruses infect a vine? And I'm going to take you down really at the cell level, at the individual cell level. As I mentioned, viruses are obligate parasites. So they go inside a cell. They do not stay on the surface of a leaf, of a shoot, or a root. They are inside. So they penetrate, and we're going to have to go back later on as to how they penetrate the cell. They have to have a hole in order to enter. Because viruses and like fungi or bacteria are so small that they cannot and do not encode all the proteins and enzymes that are needed to degrade the cell wall of a vine cell. They need to have a puncture. And very often, in relation to Ken's presentation, the puncture is made by an insect vector, when the insect vector is feeding. So the virus enters the cell, and as soon as it enters the cell, this icosahedral shape of fan leaf will just open up. You can imagine the basketball losing air and will open up so that the genetic information gets released in the infected cell. And once this genetic information is released, immediately there is a very intimate relationship that goes on between the virus and the plant nutrients, organisms, proteins, enzymes, because this is how the virus survives. It basically hijacks some of the primary functions that are needed for the plant to thrive to its own benefit, because it is so tiny that it cannot survive on its own, unlike a bacteria and like a fungi. The virus has to hijack a number of proteins in order to survive and multiply. So that's what is happening. The genetic information goes there. It is used to make the viral proteins, and then the whole thing gets together again in a very intimate way between plant constituents and virus constituents, and they form this type of factory, if you want, close to the nucleus of a vine cell where the virus propagates, basically self-clones itself by using, again, constituents of the plant. And after that, you have a multitude of little viruses or genetic information of the virus that penetrated that cell that will form, again, virus particles, and these new virus particles are ready to be picked up by an insect vector that feeds on these cells and to start a new infection of a healthy vine. So that's basically a cartoon representation of what's happening. You have penetration, you have production of proteins, you have replication of the virus, and the virus is then picked up by an insect vector and moves on. But obviously, in order to infect the whole vine, the virus has to move from cell to cell and from the cell of the organ where the infection started and invade systematically the whole plant. How does the virus do that? Because again, there the virus is facing a conundrum, because plant cells are adjacent to each other. They communicate, but the communication gaps are very, very narrow to just let nutrients flow from one cell to the other, and very tiny molecules that are even smaller than viruses. So how do viruses move from cell to cell? Well, they're basically tricking the plant. And how do they trick the plant to move from cell to cell? They produce themselves proteins by hijacking, again, some of the functions of the plant in order to widen the very narrow gap that exists between two adjacent cells. So the virus will produce these proteins that will stuck into these narrow gaps and widen the gap so that the virus particles can move through and go from an initially infected cell to a healthy neighboring cell. And once they do that repetitively over time, they will move from the initially infected cells to the vascular tissue, and through the phloem move up and start invading a whole plant. So the virus gets inoculated in a cell, opens up, proteins are produced, the virus factory produces millions of copies of the virus. These copies get encapsulated again in the basketball type shape particle. The insects can move them further. In the meantime, the viruses move from cell to cell and invade the whole plant and cause disease symptoms. So some of the diseases that is bringing us together today are leaf roll and red blotch. And here are two typical, what I would personally refer to as typical virus symptoms on Cap Franc. A red blotch to your left, leaf roll to your right. So leaf roll, you have very often a uniform reddening of a leaf blade. Very often, the main veins remain green, and you have downward rolling of the edges of the leaf blade. In terms of red blotch, you have red blotches that occur, that will start develop from the edge of a leaf blade or from the center and eventually coalesce. So this is textbook talk. Let's go to reality. I'm going to invite you to participate in a little trivia with me this morning. Are you ready? Are you game? I know Larry's game, so let's do it. So I put up on the left, red blotch, leaf roll. Just to make your job easy, I'm going to show you only Cap Franc infected only either with red blotch or leaf roll. Okay? I'm going to ask you to guess what virus is causing this type of symptoms. So if you believe red blotch, raise your hand. If you believe leaf roll, raise your hand, please. Wow, you guys are good. How about this one? If you believe red blotch, raise your hand, please. If you believe leaf roll, raise your hand. You guys continue being super good. How about now? Red blotch, raise your hand. Leaf roll, raise your hand. Right? Leaf roll. How about this one? Red blotch, raise your hand. Leaf roll, raise your hand. Again. Red blotch. How about this one? Red blotch, raise your hand. A few. Leaf roll, raise your hand. Red blotch. Okay, let's move to wide-berried wine grape varieties. In terms of Chardonnay here, overall for red blotch or leaf roll, symptoms are less conspicuous as in red-berried varieties. And you see for red blotch this interveinal chlorosis that looks like a lot like nutritional deficiency. For leaf roll, you have an overall chlorosis of the canopy and downward curling. These are textbook pictures. How about the reality here of a Chardonnay that has been extensively netted because to prevent bird damage? Red blotch, raise your hand. Leaf roll, raise your hand. Come on, guys. It is red blotch. Wow, who would have guessed, right? I was in your shoes many years ago. Blindly, I would have bet many cases on red blotch, and I was way off. Anyway, the last one. I have a series of pictures here. Only one is red blotch. Others are due to leaf roll. physical damage to a vine, nutritional deficiencies, or insect damage. Okay, you had a chance to look at this panel. Red blotch A, raise your hand. One in the back. Red blotch B. Red blotch C. D. E. The jury is still out there. F. G. Okay. The group is split. H. I. J. The person in the back is the winner. A is the only red blotch infected sample shown here. Quite amazing, huh? B is leaf roll, C is leaf roll, D is crown gall. E, you see the little arrow at the bottom right there of the picture, is girdling to a tie. You see a white arrow in the upper left corner. It's a broken shoot in F. G is mite damage, and the three others are nutritional deficiencies. Anyway, so more importantly than just foliar symptoms is obviously the effect on productivity and fruit quality. And if you can look at this comparative picture of Cap Franc, you can see that on the right-hand side, you recognize the leaf roll disease symptoms, but try to count the number of clusters. And you can see a depression in the number of clusters, so yield impact of leaf roll to the right. Same for red blotch, and this is Pinot Noir. You see the symptomatic diseased vine to your right. Look at the compactness of the leaf clusters, and more so the coloring of the berries in terms of anthocyanin in the skins and so forth. So yes, leaf roll and red blotch are very damaging in terms of yield and quality of production. And we mentioned, just to follow up on Ken's presentation, that viruses are transmitted by different insects. And again, there is an intimate relationship between a virus and a vine, and so exists an intimate relationship between a virus and a given vector. And just to emphasize what Ken said, leaf-roll viruses are transmitted by mealybugs and soft scales. They are not transmitted by any other insect species. Dagger nematodes are transmitted by fan leaf, and other related viruses are transmitted by dagger nematodes. And this is just depicting the type of insect vectors that are known to transmit viruses from vine to vine or from vineyard to vineyard. And just to illustrate, I know that Gerhard later on will provide you very valuable insights as to how leaf roll is spread in vineyard. Just want to take this opportunity and show you what we have done in terms of red blotch. And this is just an overview of an estate where there were a number of Cap Franc and Merlot in Napa County. And I was very interested in the Cap Franc in the middle there, because to the east side of the block, and you see the red dots there, I saw an aggregation of diseased vine. And at that time, I had believed, when this vineyard manager called my attention on this Cap Franc, I believed that the disease, red blotch, was emanating from the riparian area to the right-hand side there, and something was coming out from the woods and starting the infection. Well, I can tell you I was way off, because the infection started with bundles of planting material that were infected, and somehow all ended up at the edge of the vineyard there, close to the riparian area. Anyway, it's a 5-acre vineyard planted in 2008. The first symptoms were observed in the 4th leaf in 2012, and the virus was confirmed present. So I mapped that vineyard. So each cell represents a single vine. Red is symptomatic, so disease with red blotch, and white blank is asymptomatic. And we monitored with the students in the lab this vineyard over 5 years. And just for the sake of our conversation, today I'm going to split this distribution of infected vines over time throughout the whole vineyard or throughout the delineated section to the right-hand side close to the riparian area there. And this delineation is just artifactual. It's just me coming with a computer and drawing a little square around that block there. So overall in 2014, again, that's in the sixth leaf, the overall vineyard had a 4% disease incidence. And in the sub-portion there, in the subsection of vineyard, the disease incidence to start with was 32%. Now buckle up, I'm going to walk you through this vineyard from 2014 to 2018. So the newly infected vines are in green here. 2016, they're in blue. 2017, they're in purple. And 2018, they're in peach. At the bottom, I forgot to mention that the graph represents a transects of the vineyard across rows in 5 vine panels. And basically what you see is obviously a differential dynamic of red blotch spread throughout the whole vineyard, and in the section that I artificially delineated, the section where initially there were a strong aggregation of infected vine. And this is a take-home message in terms of management. I will come back later on. It is very, very key. So again, throughout the whole vineyard, just to summarize this type of work, there is a 1 to 3% increase of red blotch. And you say, ooh, what's the big deal? I can live with 1 or 3% incidence in my vineyard, which is absolutely correct. However, when you look at what is happening at the sub-portion of the vineyard there, where aggregation started the whole epidemic, you have a 10 to 20% annual increase in the number of newly infected vines. So if I summarize, viruses can be very impactful on business. And I teamed up with ag economists many years ago and your farm advisors. And these are the numbers we came up in terms of impact of leaf roll and impact of red blotch. What we know is leaf roll and red blotch, like any other grape viruses, only infect grapes. What I mean by grapes, I mean grapes in production vineyards, grapes in foundation vineyards, and wild grapes. So Vitis californica along riverbanks and so forth are also infected with red blotch and leaf roll viruses. Once a vine is infected with a virus, there is no cure in a vineyard. Okay? The virus-infected vine will keep the virus until it dies on site or it is removed. And particularly, there is no chemical that can be used against a virus. And the reason being, I'm going back to my little general statements about how viruses are obligate parasites. You can easily imagine if we were to develop a chemical that is effective against the virus, that chemical automatically would have a side effect on the plant, which obviously would not be desirable. Viruses, including red blotch and leaf roll, are graft transmissible, meaning they can infect either the sign material or the rootstock, and means that also they can be transmitted through top working. So if you top work an infected vine, the virus will move up from the roots into the scion. Okay? And this is illustrated here. And you remember red blotch on Chardonnay that I showed before, this picture on the left? So a grower didn't meet the expectations in terms of ripeness of a Cab Sauv in the Lodi area because of red blotch. And the vines were tested positive for red blotch. Unfortunately, the grower believed that if he doesn't meet the minimal requirement with a red, he might meet it with a less demanding varietal in terms of standards. So he top worked these infected vines with Pinot Grigio, and he told me, I solved the problem. I don't see reddening. Well, you would not see any reddening on a Pinot Grigio, but you can see the similarities in the symptoms, this interveinal chlorosis. So basically what has happened is from the stalk, or what is left from the cap soft, the virus moved up on the top worked vines, and now he is dealing with a Pinot Grigio infected, red blotch infected vineyard. On a bright note, red blotch and leaf roll, like any other grape virus, is not mechanically transmitted in vineyards. And I have to explain you what I mean by that. It means that hedging, pruning, harvesting, suckering, you name it, does not move a virus from an infected vine to a healthy vine, from an infected vineyard to a healthy vineyard. This being said, again to tie in with some of Ken's comments, mechanical activities in a vineyard can move the insect vectors that do carry the viruses from an infected vine to a healthy vine, from an infected vineyard to a healthy vineyard. Okay? And I'm often asked, what is the implication of insects in terms of movement of viruses, particularly with the red blotch, relative to the planting material? And I can tell you, based on my observations, my limited experience with red blotch, 90 to 95% of the presence of red blotch in a vineyard is explained by the poor selection of the planting material, not the insect vectors. What is challenging in terms of visual observations of virus presence based on disease symptoms, infection is in most cases latent in rootstocks, meaning that rootstocks can be infected, but they will not express any disease symptoms. We already talked about the role of insects in vineyard transmission, in virus transmissions in vineyard. So in terms of management, trying to put everything together, what have we learned? You talk to a virologist, he will tell you, and if you talk to most of the entomologists, they will tell you the same. Focus all your disease management efforts, the majority of it, into eliminating the virus inoculum in your vineyards. That's where you should put the emphasis. I know it's such a simple message, but you remember the disease spread over time for red blotch? I kept on telling the grower to eliminate that part of the vineyard. And obviously, he didn't listen to me. Why would someone listen to a French-American hybrid anyway, right? To start with. But can you imagine how much he would have saved in terms of the quality of the production if he just had removed that section out there? Anyway, that's his decision. And I'm fine with the decision, because a management decision is based on the incidence of the viruses, the distribution of the viruses, how this distribution might change over time within the business model of what you are carrying out. And if you decide to live with the viruses, that's perfectly fine to me, as long as you are aware of the consequences and the risks you're taking. Okay? So, in terms of management, scouting for leaf roll and red blotch right around grape picking time, or right immediately after. Flag the vines that are suspicious. Make sure you test some to know whether you're dealing with a virus, right? Remember the different slides and the trivia we just played a few minutes ago? Know whether you're dealing with leaf roll, with red blotch. And if your disease incidence is roughly 25%, rogue. Eliminate individually infected vines. And particularly make sure you avoid the infected vines being aggregated. Otherwise, the disease will go rampant very, very quickly. If the disease incidence roughly is more than 25%, you are better off removing the entire vineyard and replanting. Now, obviously, there are many considerations. These are just a guideline. Doesn't mean that you have to apply this 25% threshold to every of your blocks throughout the whole estate that you're managing. Just a guideline to keep in mind, because there are price differences, price penalties, and you know best the history of your different blocks than we do. So now in terms of knocking down, reducing the populations of insect vectors, in terms of leaf roll, as Kent mentioned, I'm very supportive of the use of insecticides, particularly knowing how efficient some of the chemistries that you have been using are, and I agree that the timing of these applications are really key to knock down and the insect population with the aim of reducing the spread of leaf roll viruses. But again, keep in mind, eliminating the virus inoculum should be the major target of your management strategies. Okay? In terms of red blotch, I do not recommend the use of any insecticides. This is because the virus population, the insect vector population in a vineyard is not abundant. This vector does not like grapes. It does not reproduce on grapes. The three-cornered alfalfa hopper hates grapes, actually. So you would have a tough time knocking down the population if it's not residing within a vineyard. Okay? And the last take-home message is be careful and spend quality time selecting your planting material. Again, just to conclude, focus your efforts on eliminating the virus. And with no further ado, I thank you so much for your attention. I want to thank all the sponsors that contribute to my research. Thank you.
Leaf roll virus for us is probably the biggest threat that we face in the industry. I mean, more than labor, more than anything. It's a huge cost, and this meeting brought together it combined the foremost authorities on the subject in the industry globally, and the knowledge they provided was fantastic.